Hey guys, welcome back to AP Biology Unit 5 Heredity. This is part 2, and let's get straight into the video. Let's get into hypothesis testing. Okay, hypothesis testing is used by scientists to reject or fail to reject. Do not say accept. You have to say fail to reject a statistical hypothesis. And it helps to determine if differences in numerical data are due to independent variables or chance. A null hypothesis states there is no relationship or difference between two groups of data. Chi-square goodness of fit test can be used to determine if there is a significant relationship between two groups of data. On the exam, you may be asked to perform one to test genetic crosses. So here are the steps to performing a chi-square test. Number 1, establish a research question. Number 2, determine the null hypothesis and the alternative hypothesis, which is just alternative to the null one. Okay, number 3, count the observed values, which is just what you see. Number 4, determine expected values, which how you do that is you get the total of the observed values and you multiply that times the fraction of the category ratio. I can explain that more in an example. Number 5, calculate the chi-square value with this formula, which you will get on the formulas reference sheet on the exam. The sideways M means you add all the values together at the end. The O stands for observed values, and the E stands for expected values. Number 6, calculate degrees of freedom, which degrees of freedom is just number of outcomes minus 1. Number 7, select the p-value. Most likely, the p-value is going to be 0.05 unless said otherwise in the question. Number 8, identify the critical value on the table. This table will also be on the reference sheet when you take the exam. Number 9, compare the chi-square value to the critical value and reject or fail to reject the null hypothesis. So if the chi-square value is less than the critical value, you fail to reject the null hypothesis. If the chi-square value is greater than the critical value, then you reject the null hypothesis. Okay, let's look at an example for chi-square testing. You can try this problem by yourself if you want to try to practice. So first of all, you can see there's a lot of text in this problem. Most of the time, you won't have the time to actually read through all this text on the exam. So just look at the graph first, and if you can, and at the actual question, which here is calculate chi-square and determine if the genes for body and wings are linked. So you just read that one sentence and the graph, and if you still can't figure it out, you skim through the actual paragraphs that are like here. F1 just means offspring of the parents. It's just a way to label generations. So the parents are true breeds. That just means they're homozygous. So one of them is homozygous dominant and another one is homozygous recessive. So that means that the F1 generation would be heterozygous. And the results on the chart we see here are F2. So the cross between two heterozygous F1 offspring. That's basically an explanation of this question, a summary of this question. So don't get too confused about F1, F2. They're just trying to confuse you here. So gray is dominant and long is dominant. So that would make ebony and curly wings recessive. So we do the cross. You would do a dihybrid cross in this situation and figure out the ratios. So we would do a cross between two heterozygous flies to come up with the ratios. And the ratio you should have gotten, which you should get this for every heterozygous cross, would be 9331. And now we're going to find the expected values. So the expected value for gray body and long wings, what you do is you simply add up all of the categories together to get the total, and you would multiply that times 9 over 16. It's 9 over 16 because 9 plus 3 plus 3 plus 1 equals 16, and 9 out of these 16 is gray body and long wings. And you would just do the same thing for the other 3 categories, and at the end you would use the chi-square formula and add all the values up together to get the chi-square value. Since it doesn't tell you a p-value here, you would just go with 0.05. Look at the table. We need to find degrees of freedom. So the number of outcomes here is 4 outcomes, because you can see on the table there's 4 slots. And you would do that minus 1, and you would get 3. So the degrees of freedom is 3. You would look at 3, and you would compare the critical value on the table to the chi-square value and figure out if the genes for body and wings are linked or not. So the chi-square that I calculated was 2.7556. So we compare this chi-square value to the critical value on the table, which is 7.82. And obviously the chi-square is going to be less than the critical value, so we fail to reject the null hypothesis. So these genes, the genes for body and wings, they have no relationship and they are not linked at all. Okay, now that we finish with Mendelian genetics, let's get into the 4 cases that genetics will not follow Mendel's laws. Linked genes are genes that are close to one another on the same chromosome and are usually inherited together. They are less likely to be separated during meiosis, and it's important to know how to calculate the map distance, which is how close a pair of linked genes is, and also how frequently a pair of genes participates in a single crossover event. An example of this is linked genes with a recombination frequency of 5%, and this means the genes are very close on the chromosome. As you already know, humans have 46 chromosomes, and the last pair of chromosomes determines sex. In males, that is XY, in females, that is XX. Sex-linked traits are traits that are determined by sex chromosomes. In humans, traits are carried by the X chromosome because the Y chromosome only codes for maleness. There are carriers, which are heterozygous genotype of the recessive sex-linked allele. In pedigrees, carriers are shown as a half-shaded shape. Traits determined by multiple genes also do not follow Mendelian patterns. An example of this would be human hair color. Some traits that are inherited are not stored in the nucleus, and these traits would be chloroplasts and mitochondria because they have their own DNA separate from the DNA in the cell's nucleus. In animals, mitochondria are inherited from the egg cell and not the sperm because the egg cell is bigger and contains the organelles. In other words, you get your mitochondria from your mom. The same genotype can result in multiple phenotypes. This is because environmental factors can influence how genes are expressed. Examples are calorie intake can change your body shape, and time in the sun changes your skin tone. Phenotypic plasticity is the ability for one genotype to produce more than one phenotype. Human genetic disorders are caused by inherited mutated alleles. Mutated alleles can be randomly distributed and carried by chromosomes. Mutations that lead to genetic disorders can negatively impact the offspring. An example is blindness. Not all mutations are negative, though. Some mutations can be neutral or actually beneficial. Human genetic disorders caused by chromosome abnormalities. So the vocabulary word you should know for this is non-disjunction, which is the failure of chromosomes to fully separate during the formation of gametes, or the egg and the sperm, and it leads to too many or too few chromosomes in the sex cells. An example is XXX in females. Mutations or misformations in sex cells / gametes can result in disorders being present in the offspring that were not present in the parents. Okay, so I've covered all of the important material for Unit 5. Check out the Selfie website for my notes in each lesson. The link is going to be in the description. Subscribe for more AP Biology content, and thank you for watching.